Welcome to the External Medicine Podcast. I'm your host, Mitch Belkin, and this is my co-host and brother, Daniel Belkin. We are here with the theoretical and experimental immunologist, Polly Matzinger. Polly received her PhD in biology from UCSD before completing a postdoc at Cambridge. She then worked at the Basel Institute for Immunology before moving to the NIH, where she was a section head at the National Institute of Allergy and Infectious Diseases of the so-called GHOST Lab. Polly, welcome to the show.
Thanks. Nice to be here.
Before we get started, do you have any financial disclosures?
I am part of the NIH. You're not allowed to do any of that kind of stuff. I have a farm, but I don't think what we're doing here will impact the income of the farm.
Well, speaking of your farm, you have a lot of extracurricular interests. You've done a lot of things. Your CV includes things such as bartender and dog trainer and jazz musician. Can you talk a little bit about your background and then how you eventually got interested in science and became a biologist?
Sure. So I started off wanting to be a composer. And so I did music. And after 2 years of college music, I realized that the music I write is dead boring. Even to me, it's dead boring. So I was going to end up teaching high school choir, or something like that, and didn't think that was particularly exciting. So I quit. And did a lot of other stuff and enjoyed them. But after a while, began to think that everything gets boring after a while. So I switched. I did all kinds of stuff. Did, carpentry for a while and training dogs for a while. I still do that, but you never stop doing that. Being a Playboy bunny was fun. Lots of, and lucrative as well. And then I decided that I was just going to be a barmaid. I'm good at it. I made decent money at it. And I could work nights and do what I wanted with my days. And so I was working in Davis, California, at a place called Mr. B's. And every week, a couple weeks, two professors would come in and get a pitcher of beer and talk science. And I would listen to them. And one day they were talking about mimicry. How viceroys mimic monarchs because the monarchs taste bad. And so viceroys look like them and the birds don't eat them, and like that, stick insects and stuff. And I asked them a question that had bothered me for some time, which is, why has no animal ever mimicked a skunk? You would think that a raccoon with a white stripe down its back or a cat with a white stripe down its back would have a selective advantage. And their mouths fell open. Cocktail waitresses don't ask these kinds of questions. And they didn't have an answer. But they said they'd think about it and come back. And they came back, a week later, and we talked about selection pressures and population generation time. But there was no answer. Anyway, long story short, lots of other things in between. One of them, Professor, what was his name? Swampy, everybody called him, decided I should be a scientist. By that time, we'd had lots of conversations about rabies vaccines, about all kinds of stuff. And I blew him off. I said, nah. Everything gets boring. That's going to get boring. At least with this job, I get to do what I want with my days, da-da-da. And he didn't give up. That man worked for 9 months. He would come into the bar. He'd bring me an article about all kinds of stuff about magnets in pigeon brains, about communication in electric fish, from Nature, from Science, from Scientific American, right? And he'd say, read this. And he'd come back 2 days later as the bar was closing, and we'd go to a Denny's and talk about the paper he'd given me. He did this for 9 months until I finally said, okay, I'll give it a try. I've tried lots of other things. I'll give this a try. I owe that man my career. Anyway, he helped me apply to graduate schools, and I got in and started in biology. Did Drosophila genetics And UCSD has a really interesting, or did at least in the 70s, program where, like a lot of other PhD programs, you have to do a preliminary exam. But unlike a lot of other PhD programs, you have to do your preliminary exam in a topic that is totally unrelated to your PhD topic. And my PhD topic was about sex determination in fruit flies. And my roommate's boyfriend was an immunologist. And he said, you should do MHC restriction. This was in 1977. He says it's fascinating. There isn't much literature because it isn't very old, so you don't have to read a lot. And it's totally unrelated to sex determination in fruit flies. So I started reading about that. And the argument that was going on then was, do T cells have one receptor or two? And I decided it had one. And Rod Langman, this boyfriend of my housemate, said, no, they have two. And we had all kinds of arguments. And in the middle of one of the arguments with him, I had my first creative thought. And when you have a creative thought in a field, having tried to be creative in music and failing abysmally, right? When you have a creative thought, you switch to that field. So that's how I became an immunologist. It really is, I get by with a little help from my friends. Somebody talked me into going back to graduate school at age almost 30. Somebody in the graduate school talked me into studying a subject I wasn't interested in. And here I am.
What was your creative thought in that conversation?
So the question was, why do so many lymphocytes respond to foreign MHC? Right? This is the biggest barrier to transplantation. You have to match MHC, HLA in humans, right? For most antigens, the frequency of lymphocytes that recognize it is, 1 in 100,000, 1 in a million. For MHC, it can be up to 10% of all your T cells. So why is that? It was a huge question then. And remember, this was before we knew that the MHC captures peptides and takes them up to the surface of a cell. We didn't know that then. But we somehow knew that MHC restriction had just been discovered. So we knew that the MHC had something to do with being a target for T cells that see other antigens. So here was my creative thought. The reason the frequency is so high is that the T cells are not responding to MHC alone as a single antigen. They're responding to MHC in combination with 1,000 other antigens that the cell puts up on its surface. That was the idea. It's not one target, it's 1,000 targets. And because it's 1,000 targets, you have a high frequency. Pretty simple. Very controversial at the time. I remember going to a meeting and having Jack Strominger, whose lab did discover that MHC has peptides in it, pointing at me, a graduate student, in the audience saying, "You're wrong." So, yeah, I've been swimming upstream since the beginning.
Let's get into the danger theory, which is sort of one your theory of immunology that sort of puts together all these different pieces that you were working on and thinking about over the years. But before we talk about the danger theory, can you just walk us through some of the previous theories of immunology, the self, non-self theory and the Janeway theory, and then we'll get to the danger model.
I really wish I had a diagram I could write, because so when I give lectures, I do this thing that I call 70 or 80 years of immunological history in one diagram. So it starts in the early 50s in Australia with Macfarlane Burnett. And so people were trying to understand how you can make antibodies to things that didn't even exist in nature, things chemists were making in their labs, and how that was controlled. And there were lots of theories. And Burnett modified a theory from Niels Jerne and suggested what is called clonal selection. So B cells that make antibody, B cells which make antibody. Burnett said each B cell has on its surface a copy of the antibody it's going to produce. And when that antibody binds antigen, it stimulates the B cell to divide and make antibody. Right? So that's what we call today signal one. The B cell sees its antigen, it gets stimulated, it divides, it makes antibody. And the idea was that early in life, you kill off all the B cells that can see yourself. So all that's left are B cells that can see foreign antigens. It was the beginning of a self, non-self model. And a guy named Peter Medawar did an experiment where he injected baby mice with bone marrow cells from a foreign mouse. And indeed, when those baby mice grew up, they were able to accept skin grafts from those foreign mice. So the idea was that early in life, you have this period where you learn what self is. And then everything after that is non-self. Right? Transplants are non-self, viruses are non-self, bacteria are non-self, et cetera, et cetera. I got to tell you, when I was a graduate student, I had terrible trouble with that idea. I asked, what about fetuses? They're non-self. Why don't pregnant women reject their fetuses? And Medawar worried about that too. And he came up with some pretty amazing ideas, which we'll discuss later. I asked, what about the newly lactating breast? It's making proteins that body has never made before. And they're being presented, and the immune system should react to them. And my professors patted me on the head and said, there, little girl. Babies drink mother's milk and become orally tolerant during that early period. And I said, you know what? My generation's done an experiment. When I was born, it wasn't cool to drink mother's milk. You drank Nestle's Carnation. Those companies are still trying to do this to third world women. But the important point was that when my generation grew up and started lactating, our breast didn't fall off. The immune system somehow knows how to deal with a changing self. But I had to put all that aside because I had no answer. So that was the beginning of wondering about this whole self, not self business. All right, back to Medawar. Mel Cohn at the Salk Institute and Peter Brecher were worried about something that had just been discovered, which was B cell hypermutation. So when B cells respond to an antigen, not only do they divide and start spewing 2,000 antibody molecules per second into the blood, they mutate. In fact, they really mutate, they hypermutate. And the idea is that B cells that have low binding affinity to an antigen, by mutating, might get higher binding affinity, and thereby your immune response gets better with time. But it causes a problem, because if you get rid of all the self-reactive B cells early in life, and now your B cells start mutating when they see flu, right? You're going to make some B cells that see self. And so Peter Brecher and Mel Cohn were worried about this, and they pulled a model out of the air. They said, the way you can fix this is to add a second cell. They called it a helper cell. They didn't know about T cells yet. They called it a helper cell, and they said, if you require for an immune response that two cells respond, two different cells respond, you can fix this. So it would be rare that cell A and cell B, that both see the same antigen, flu, would mutate to see the same self antigen. And so he said, all right, you have the cell that responds and you have the cell that's going to help it. And they said, if you really want to fix this, you need 3. However, since the frequency of cells responding to any antigen is 1 in a million, you would need 1 times a million, times a million, times a million numbers of cells. And we don't have that many cells. So they settled on 2. This is how immunological theory happens. And now we know they're right, that a B cell that doesn't get help from a T cell helper cell doesn't respond. So that was 76. No, that was 1969. And then back in Australia, Kevin Lafferty was worried about another phenomenon, and that was, why is it that T cells from one species respond more strongly to individuals from the same species than to other species? So T cells from a human will respond very, very strongly to a foreign human. And dogs respond very strongly to a foreign dog, and so on. But mice don't respond very strongly to human cells, and human cells don't respond very strongly to dog cells, and so on. And yet you would think that a dog was more foreign to a human than another human. So he fixed that by adding another cell. You see, there's a pattern coming here. He added what we call today the presenting cell. He said T cells themselves need a second signal. So when they see antigen, they will die unless they get this second signal. And they get the second signal from this accessory cell, the macrophage. Right? And that model was totally ignored for 13 years. Until it was rediscovered by accident by Ron Schwartz at the NIH. When they were trying to find out how MHC presents antigen. And so they were taking macrophages, giving them antigen, glutaraldehyde fixing them, and then offering them to T cells. And their idea was that they could use antibodies to pull down the MHC and find out what that MHC is hanging onto. But it worked. They discovered it didn't work. The glutaraldehyde fixed macrophages did not activate T cells. And so they said the T cell needs a second signal. And they actually referenced the wrong model. They referenced Belkin. Anyway, now we know that not only do B cells need a second signal from T helpers, T cells need a second signal from antigen-presenting cells. Right? So we're adding one more cell to the immune response each time. So the question was, why was the macrophage ignored for so many years? And here's what I think was going on. If it's the presenting cell which controls whether you start an immune response or not, you can't have a self-non-self model. If it's the B cell that makes that decision, you can, because you can remove all the B cells that see self. If it's a T cell that controls that decision, you can, because you can remove all the T cells that see self. But macrophages aren't specific. They pick up everything. They present everything. So if the dendritic cell, we'll bring to dendritics. If the presenting cell is what controls whether you respond or not, and it presents everything, you can't have a self-non-self model. So it was ignored for a very long time. And when it was finally rediscovered by accident, it really put a monkey wrench in the works. And 3 years after it was rediscovered, Charlie Janeway offered a solution. He said the presenting cells themselves also make a self-non-self discrimination, but it's a different kind. It's an evolutionarily selected one, that presenting cells respond to conserved molecules on evolutionarily distant organisms like bacteria. So without that signal, the presenting cell doesn't turn on. And without turning on, it doesn't give a second signal to a T cell. That's the most important thing he said. And people have kind of ignored that. Up until that time, it was thought that macrophages were constantly presenting what they found to T cells and turning them on. No, they're quiet. They're totally quiet. Well, probably not totally quiet, but they're quiet as far as having a second signal to give to a T cell. And Charlie said you have to turn them on. And what turns them on are molecules from bacteria. PAMPs, pathogen-associated molecular patterns. And that's all very well and good. It explains how you respond to bacteria. But it doesn't explain how you respond to viruses. It doesn't explain why you reject a transplant. You know, most well-done heart transplants are not covered in bacteria. It doesn't explain how you get autoimmunity or allergy. So the model still needed something. So Charlie took the first step away from this pure self, non-self, but it needed something. And when I pointed that out to Charlie at a meeting, he said, no problem. Transplants are a modern invention and the immune system doesn't have to deal with it. Okay. And autoimmunity and tumors kill you late enough in life that you've already had your kids, and so evolution doesn't care. And I said, you know what? You have to explain what the immune system does, not just what you happen to think it evolved to do. And it does reject transplants, and it can get rid of a tumor, and it does give you autoimmunity, etc. So you need something to figure that out. You need something more. And I didn't know what that more was going to be. But there was this really brilliant postdoc in Ron Schwartz's lab named Ephraim Fuchs, who's now at Hopkins working on cancer. And Ephraim was also beginning to question the self, non-self model. And I, by that time, was in old fogey mode. And so we'd have these arguments. I was defending the model. He was arguing against it. And we probably argued for 2 years before, and neither one of us can remember who first came up with this. The idea that the immune system responds to danger rather than to non-self. We came up with that idea after a couple of years of arguing. So all well and good. How does it know what's dangerous? That took another year. And that happened to me in the bath. I used to take long baths until I had a farm. I used to take long baths and sit in the bath and think and read and write. And one day, bang, it came just like that. It was very, very simple. Something is dangerous if it does damage, and it's not dangerous if it doesn't do damage. And as Ephraim would put it, if a virus comes in and makes 3 copies of itself and leaves, you don't have to make an immune response. You might even welcome it because it could bring you genes that you wouldn't get otherwise. So, okay, we've taken one more step. How does the immune system know that damage is happening? Was the next question. And that happened to me while I was training a dog to herd sheep. So it was July, it's hot. Sheep have woolly coats. You can train for about 10 minutes and then you have to take a break and let everybody cool down. And so we were taking a break. My dog and I were sitting on this little hill and there were sheep at the bottom and there was a stream and a little bit of woods behind. And all of a sudden, this sheep comes running out of the woods going, meh, meh. They do meh, by the way, not bat. And my dog took off into the woods. And I thought, that's it. There are these cells that are like sheepdogs. They're curled up asleep, noses under their tails, called dendritic cells, and they're normally asleep. And it's the damaged cell that tells them that damage has happened. So my dog had not run into the woods because she heard something into the woods. She ran into the woods because she heard this sheep come running out of the woods, scared shitless. It's the sheep that tells you it's the damaged cell that tells you. And so we suggested, Ephraim and I, that damaged cells send alarm signals that wake up APCs, and that you don't get an immune response normally until you wake up one of those APCs. And what wakes them up are signals from damaged tissue. And that is the fundamental danger model.
Fascinating. Can we walk through just a very brief outline of what normally happens during an infection, just to introduce this cast of characters? You mentioned B cell and T cell and antigen presenting cell. So let's say you get an infection of some sort. What is the classic understanding of how the body responds to it? Just so that we can introduce, we can go into the detail. Yeah.
The classic or the new?
Well, what happens? Like,
okay,
if you
so
get,
here's what I think happens.
Okay.
And by the way, when I teach people who are really not immunologists, I don't use the word antigen. So let's actually define antigen. Where did that word come from? It came from antibody generator. So an antigen is something that generates antibodies. And 1971, Niels Jerne. And immunologists distinguish between antigens and immunogens. So an antigen today is not something that generates antibodies. It's something that can be a target of antibodies. And that could be really any shape. An immunogen is something that is both an antigen, that it can be targeted by antibodies or T cells, and by itself elicits an immune response. Because not all antigens do that. Right? If you want to make a vaccine and you use, say, a modified live virus, all you have to do is use the virus. But if you want to make a modern designer vaccine, proteins, peptides, whatever, you need to add something that is actually going to give you an immune response. This is what Charlie worried about. You can't just inject TNP and expect to get an immune response. You can't just inject a peptide and expect to get an immune response. You don't. You need something to activate the antigen-presenting cells. So here's what normally happens in an infection. Let's take flu. So flu comes in, it infects some epithelial cells. The epithelial cells send alarm signals like type 1 interferons, interferon alpha, interferon beta. Destroyed epithelial cells also release their DNA, their RNA, their mitochondria, which are, by the way, internal bacteria, which brings Charlie's model and my model together, because they are bacteria and they're inside cells and not normally found outside cells when the cells are healthy. My cat is snoring. Shall I do something to stop him? Okay. So the flu comes in, it damages epithelial cells. The epithelial cells send alarm signals that they elaborate, like type 1 interferons, or when they're smushed, they release DNA, RNA, and other things which also act as alarm signals. Those alarm signals are picked up by the local dendritic cells, which until that moment are quiet. To a point, we'll come back to that. They leave the area of the infection and go to local lymph nodes. They get turned on, and there they chew up what they have picked up and present it on the surface in MHC molecules, and the T cells going by scan them. And if one of that 1 in a million or 1 in 100,000 T cells that's going by binds to those presented antigens, they get signal 1 by binding and signal 2 from the activated dendritic cell. And that tells them to make an army, turn on, and after 3 or 4 days of dividing, they leave and go looking for the infected tissue. So damaged tissue, alarm signal activates the APC. The APC chews up the antigens and presents it on its surface. T cells going by see it, get turned on, make an army, and go out looking for the infected tissue. And a killer cell can kill one infected cell in an otherwise healthy tissue. They are the world's best microsurgeons.
If that's the case, that your friend who you worked on this theory with said that a virus that comes in, makes a bunch of copies of itself, and then leaves without doing any damage, doesn't get seen by the immune system, like, why don't more viruses and bacteria do that? If that would enhance their longevity in some way, why wouldn't they live a lot longer? Why wouldn't they prefer to do that and not cause any damage?
They do that. We have more and more retroviruses being discovered all the time. And you take Epstein-Barr virus. 90% of the human population on the planet has Epstein-Barr virus. Most of us never notice it, really. We might have a cold when we get it. It induces a small immune response, and then it goes and hides in B cells. So how many more of those are there out there that we don't know about? Right? We worry about the ones that don't do that. And when you think about it, some of the ones that don't do that, some of the ones that induce immune responses are evolutionarily new pathogens to us. Herpes virus. What does it do? It comes in, it infects a nerve, it quietly goes to sleep until something happens to your body that makes it think, oops, this body is about to leave this planet, like sunstroke or another virus infection, and what does it does then? It comes out, it makes a sore, which doesn't make a scar, right? And it comes out where people meet each other, lips and genitals. It's a way of getting into the next person. If we look at the plague, the first time it came through, it killed a huge percentage of the population. The next time it came through, it wasn't so bad. I mean, COVID is doing the same. It was pretty horrible in the beginning. And now it looks like even in unvaccinated people, it isn't as bad as the original. It's in the virus's interest not to kill us. It wants us walking around coughing, right? It is in tuberculosis' interest not to kill people. It wants you walking around spitting. Yeah, malaria doesn't want to kill you. Mosquitoes don't bite dead people. It wants to make you hot and slow, because mosquitoes don't fly very fast and they seek heat. And so they do that. I think the ones that make us sick are the ones that haven't, by chance, had the right mutations yet in their evolution to figure out how to not make us very sick.
So in the simplified model that you're describing, we have damage, which sets off alarm signals. The antigen-presenting cell travels to a lymph node, meets up with a T cell, which become activated, and some of the natural killer type cells can go and attack tissue. There's also the B cell response. So immunology, I should mention, was my worst subject. So correct me if I'm wrong.
Doing well so far.
T cell, the activated T cell can then activate a helper T cell, can activate a B cell, which can start producing various types of effector responses. So, for instance, can produce lots of IgG or IgE or whatever the case may be. They can produce some sort of antibodies. Is that correct?
Yes. Now, I want to make one correction. There are killers and natural killers, and they're not quite the same. The killers we know of, called cytotoxic T lymphocytes, see peptides in MHC. They see the surface of a virus-infected cell. And you might ask, why this thing about MHC? Why do these things come to the surface of a cell? Because viruses want to hide. They don't want the immune system to see them. And so in this arms race that the immune system has made with the viruses, they figured out how to prevent viruses from hiding, because the MHC molecules present peptides from whatever is going on inside the cell. It's a window display, so it's not 100% like any window display, right? But if the virus is inside a cell trying to make more virus, those pieces are going to get stuck on the surface of the cell so that a killer can see them. And the killer can see them within 20 minutes of the virus infection. So it can kill that factory of viruses before the viruses get to completion. All right, but let's go back to B cells. So a B cell will also see the antigen, the flu. But if it just sees the flu and it doesn't get help, it dies. That's part of the process of self-tolerance, right? The ones that mutate to see self won't get help. So you need those activated helper T cells to get the B cell to make antibody. And the T cells are part of what determines what kind of antibody the B cell makes. And to go one step further, I think the tissues tell the T cells what to tell the antibodies. So if you have an infection in the mucosa, you'll make IgA. It's the antibody which gets secreted into saliva, lung mucosa, vagina, etc. It's right there at the entry point. If you have an infection in the skin or a muscle, you tend to make various forms of IgG. We might come back to that with COVID, actually. But it's the tissue telling the T cell, and then the T cell telling the B cell what kind of antibody to make. I mean, I think really what controls our immunity are the tissues. They send alarm signals that activate an immune response. They tell T cells what kind of immune response to make. And by presenting on their surface their antigens in the absence of alarm signals, they allow T cells to get signal 1 without signal 2 and die. So they're constantly inducing tolerance to themselves. Is that clear, or should I do it again?
I think that's clear.
Good.
So there are, from this danger model, a lot of different potential implications. I mean, what's the value of a model? It can help explain things that exist in the real world that are otherwise unexplainable. Some of the things that you bring up are autoimmune diseases, transplant, rejection, the placenta. What can the danger model help explain that previous iterations of immunological models aren't able to?
Lots. More than we can do in this hour. So let's start with the things that worried me as a graduate student. Right? Why doesn't the mother reject the fetus? I mean, in fact, that's the first problem that the self-non-self model had. Why doesn't the mother reject the fetus? And the suggestions, which are still in the textbooks from Medawar, Nobel Prize winning guy, smart guy, doing his very best to try and explain the biology in the context of this model, suggested that the placenta is both an immunological and physical barrier between mother and fetus, and the immune system can't get through. Okay? That was one. And the other one was that the fetus immunosuppresses the mother. Now, that's a pretty stupid idea, right? You take the individuals that are carrying the next generation and you turn them into AIDS patients, right? They're immunosuppressed. Excuse me? Wouldn't be very good for the species. And it's not true. I mean, pregnant women are incredibly healthy, most of them, right? What about all those sheep out there? They're not having to live in bubbles. When I talk to people, I always ask people to raise their hand if they've had a pregnancy. And I ask them, did you have to live in a bubble? No. It's dumb and it's not true. I have found that most things that seem evolutionarily dumb are not true. So yes, there are some parasites that take advantage of the pregnant state. Malaria is one of them. There is one malaria variant which takes itself to the placenta. It's a great place for malaria. There's a lot of blood, a lot of red cells for them to get into. But it's bad for the fetus and the mother. And In fact, malaria is a kind of proof that an individual can make a primary immune response during pregnancy, that it works really well. Because women that have had a pregnancy and malaria and do badly, the fetus is low birth weight, they sometimes die, et cetera, et cetera. By the second pregnancy, they're doing better. And by the third pregnancy, they're totally fine. And a guy named Lars Hvid, Hvid, studied in Africa the antibody responses to this one variant in African villages and discovered that every woman who's had a pregnancy makes antibodies against it, and no men. Because the variant can't infect men, it goes to the placenta. So there are pathogens that have learned to take advantage of the pregnant state. But in general, pregnant women are perfectly fine. Okay? So that's the first one. The fetus immunosuppresses the mother. The second one was there's this barrier between mother and fetus. That we know now that's not true. There are fetal cells that get into the maternal circulation. And you can bleed women and run their cells and find the fetal cells and analyze them as a way of doing a non-invasive prenatal diagnosis. There's also traffic from mother to fetus, right? 50% of human kids, the kids who do have to live in a bubble, have T cells from their mothers. Not very many, but some. In fact, there's this great anecdote in transplantation from Euro transplant in the Netherlands, where they have people waiting for kidneys. And mothers are often donors, living donors for their kids. And they discovered when they test the immune response of the potential recipient to the cells of their mothers, 25% of them don't respond. They're perfectly tolerant of their maternal cells. And Lee Zhang and Rick Miller followed that up in Canada and discovered, using mice, that 25% of adult mice are carrying cells from their mothers. 25% of people are carrying cells from their mothers. So there's traffic in both directions. So what does the danger model say? It says that as long as that fetus is healthy, it's not sending alarm signals. And as long as it's not sending alarm signals, you will not reject it. Pretty simple. So people say to me, what about Rh disease? Why is it that an Rh positive fetus in an Rh negative mother has a problem? So God is in the details. The first pregnancy isn't a problem. It's the second pregnancy that's a problem. You get primed at birth. Birth is dangerous, right? You have ripping and bleeding and surgeons with knives. And that's when a mother gets primed. And we know that. And in medical practice, we know that because we give that mother antibodies to block the Rh antigen at birth. Now, occasionally you get a woman who starts making these antibodies a little earlier. And Liz Bonney, who is a gynecologist who used to be in my lab, says there are women that have what are called fragile placentas, where the fingers break off in the lacunae of blood. And that breaking is not normal death. That's dangerous death. And so you get alarm signals. And that's why those mothers, she thinks, make earlier anti-Rh antibody. But in general, danger model says as long as that fetus is healthy, the mother's not going to reject it.
So basically it's saying that it only presents an issue if there's tissue damage. If there's no tissue damage, if there aren't DNA and hydrophobic portions of cells that are being exposed to the immune system, then the immune system doesn't think that there's any issue going on.
That's right. I'm going to expand that a little bit and say anything that is not normal physiology could elicit an immune response. So it doesn't have to be a cell breaking open, for example. We know that cells do things like upregulate heat shock proteins when they're stressed in various ways. And heat shock proteins are alarm signals. HSP70, HSP60, they are alarm signals. So certain forms of stress can also do it.
What are the implications of this theory for transplants?
So why do you reject a transplant? Where's the damage? Where's the danger in a transplant?
From the surgeon.
Yeah, surgeons are dangerous. Yeah, they cut, they burn small vessels, they put organs on helicopters and fly them around the country. They cool them. This is all heat shock, right? It's cutting tissues. It's damage. And it shows up in medical practice. If you have a kidney transplant, a living donor kidney will do far better, even if it is totally mismatched, than a matched cadaver kidney. And I think part of the reason for that is that the matched cadaver kidney gets on a helicopter, or at least gets into a solution that is cold, anoxic, right? There's no blood cells carrying. So the cells become anoxic. They upregulate, in fact, this is known, they upregulate enzymes to capture oxygen. When you transplant them now, doing more damage, into the recipient, all of a sudden they get warm because the blood's coming through. They're getting oxygen, and they get what's called post-ischemic reperfusion injury. And it's the fact that they've upregulated all these enzymes to capture oxygen, and then all of a sudden they've got all this oxygen. And so they get oxygen radicals, and that does a lot of damage. So the living donor kidney, both people are in the operating theater. You take it out of one and you put it in the other. Much less damage. And they do much better. So prediction is, if you could do a transplant without damage, it would take. Right? The other thing is, if you could block the alarm signals, you might be able to get it to take. The problem with that is there are too many alarm signals. This is something so important, you don't leave it to one, right? DNA, RNA, SNRPs, mitochondria, they look like bacteria. Mitochondrial DNA is an alarm signal. Some of the peptides mitochondria make are alarm signals. So you can't do a transplant without doing damage. But one thing you could do is to block the signal that an activated APC gives to T cells. And Alan Kirk did that. Commander Kirk, actually. He was in the Navy. He's a star. You should interview him one day. He went from being a tuba player to a star transplant surgeon. He blocked signal 2. He blocked the signal that a dendritic cell gives to T, one of them, that it's a T. And he did this in monkeys. And he published a paper in Nature Medicine showing that if you blocked this signal, you could give that drug for a couple of months, I think it was 5, and then stop. And the kidneys would not be rejected, which is pretty amazing. So yeah, so it's the damage. And, I think the transplant community has pretty well accepted this. So they try to do less damage. People doing bone marrow transplants, for example, do not prepare their recipients with so much damage. They try to do lighter preparations. They try to be nicer to the organs they're transplanting. But they need to block signal 2.
2 questions. The first one is when you say signal 2, that's CD40 ligand?
CD40 is one of them. Mm-hmm. There's more than one. So CD40 ligand is what Alan Kirk blocked.
Okay. And has that been successfully done or tested in humans?
Well, yeah. My computer says low battery. Yes. But it turns out that platelets also have it. And when you give the antibodies, you cause some platelet aggregation in people that have atherosclerosis and some other diseases. And so that Alan Kirk's trial in transplants was stopped for that reason. And I don't know where he's gone with that, but you should interview him. We need to stop for a minute because I need to get a cord for I had 100% battery when we started. I don't understand this. All right. Let me make sure it's working. Okay.
Perfect. The other thing I was interested in was, so you could stop the CD40 or a second different signal too to prevent activation of the immune system when exposed to this damage. You describe an experiment with mice in which, prior to the formation of I think it's like mice that don't have a functional immune system, that you later reintroduced having an immune system. Can you talk a little bit about this experiment?
Yeah, Colin Anderson did this in my lab. And he and I disagree about the interpretation of the result, actually. But this is what he did. He took mice that had no immune system, the boy that lives in the bubble, and he gave them a heart transplant from a different strain of mouse. And if that mouse had had an immune system, it would have rejected that heart in about 20 days. But it doesn't, so it doesn't reject. So the heart heals, and by accident and so on, we discovered that it has to heal for a long time. So he left it for 9 months, and then gave the mouse back an immune system and asked, are the hearts rejected? And the answer is no. If he only leaves them in for 1 month and then puts in the immune system, they are rejected. So the way we interpret that is that the heart has healed, and it takes a long time for it to completely heal. And in fact, if we measure by PCR, the amount of healing and stress molecules that are up, it's up for a long time, 5 months at least. I now think that no organ heals until it becomes reinnervated. It's never completely healed unless it becomes reinnervated. But I'm hand waving there. People sometimes ask me if my wrists get tired of hand waving. Anyway, if you leave it long enough, it heals and it's not rejected. And even if you then try to vaccinate the mouse or immunize the mouse against the heart, it's still not rejected. So the heart has learned to tolerize for itself. It heals. If you let it interact with the immune system for a couple months, it induces tolerance to itself. Now, when Colin repeated that experiment with different strains of mice, his results weren't as good, meaning that it wasn't 100% of the mice. It was only about 50% that became tolerant. So we have discussions about why that is.
What exactly is the process of tolerance that's taking place with the heart tissue from a different mouse?
Exactly. Shorter answer, I don't know. Long answer, slightly longer answer. Here's what I think. That if the tissue is healthy, it's doing two things. One, it's presenting its own antigens on its surface in MHC, that window display, without alarm signals. So it's not activating its local APCs. And if a T cell then, T cells go through tissues, they wander through, and then go back into the blood, then, back into the tissue. They circulate from the blood to a tissue about once every 24 hours. And so if they're wandering through a tissue and that tissue is healthy, and that T cell can see that tissue, it gets signal 1 without the second signal, and so it dies. So that's one way. The other way is that, remember I said the dendritic cells, the APCs were completely quiet? No, they're not completely quiet. They are still drinking their environment. And so they're drinking their environment and putting what they find up on the surface, but they're not activated to give a second signal. So now a T cell going through and seeing a tissue antigen on a dendritic cell will also get signal 1 from binding, but it won't get a second signal, and so it dies. So there's these two mechanisms. I think there's a third, but I don't know yet if it has to do with inducing T cells to die, inducing deletion, or whether it has to do with telling T cells what kind. Remember, I said T cells tell B cells what kind of response to make. So there are what people call checkpoint inhibitors. These are checkpoint PD-1, PD-L1. So tissues make other molecules that influence what T cells do. And I don't know how many of them are involved in telling the T cell to die, or how many of them are involved in telling the T cell, we need IgA here, or we need IgG4 here. Right? But tissues are incredibly important signalers to T cells to tell them one thing or another. So it's the tissues that induce their own tolerance.
How does your theory apply to cancer? Speaking of PD-1, L1, and PD-L1 inhibitors.
Yes. Well, why don't they work in more than about 20% of people? So I have an answer, I think. A potential, hand waving, So what's wrong with a tumor? What makes a cell a tumor? What makes a cell a cancer cell is that it has lost growth control. It's dividing when it shouldn't. So lots of cells need to grow. You cut yourself, the epithelial cells need to fill that, and they start to grow. And then when they meet each other, it's called contact inhibition, they stop growing. But a cancer cell that no longer has contact inhibition, it just divides or multiplies, since multiplication and division are the same in biology. Growing is not an alarm signal. The immune system cannot discriminate between rapid cell division in the gut, rapid cell division in the bone marrow, rapid cell division in the thymus, right? Rapid cell division is not an alarm signal. So it used to be thought that we would get a tumor every few minutes and the immune system would get rid of them. And the ones that grow out are the ones that have learned how to escape. I don't think that's true anymore. We have molecules like P53 that myoblastoma that monitor this and tell cells to die when this kind of stuff happens. We don't get a tumor every 3 minutes. We get them rarely. And when they happen, they're not sending alarm signals. So what happens to a cell that's growing that is not sending alarm signals? What happens to a T cell that can see that? It gets signal 1 without signal 2 and dies. And so as the tumor is getting bigger and bigger and bigger, it's inducing tolerance to itself. Now, when they get big enough, if they're poorly vascularized, they start to die in the middle. But tumor pathologists call this sterile necrosis. It's programmed cell death. Starvation, which is what they're dying of, right? Not enough blood, not enough nutrients, is programmed cell death. Still no alarm signals. They get scavenged by local scavengers, because there are scavengers to pick up dying cells, yeah? So it's only when the cell death gets big enough, bad enough, that you overload the local scavenging capacity, that now these cells that die don't get scavenged, they fall apart. It's called secondary necrosis. Now they send alarm signals. By that time, you've got 2 T cells left that can see that tumor. And they may get activated. And each one, say it turns into a killer, each one will become 30 to 100 killer cells. And each one of those killer cells, it has been estimated, can kill 10 to 100 targets. But it takes a few days for that to happen. right? So let's say it happens. You've got enough damage now in the tumor that you overload the scavenging capacity. You turn on some APCs. They go to the draining lymph node. They turn on the 2 T cells you have left. And they valiantly try to get rid of that tumor. But it takes 3 days for them to get activated. It takes 4 days before they leave the lymph node. Now you've got 200 of them. They go to the tumor. They each kill 10 to 100 targets, and then they have to be reactivated. This takes another 3 days. What's the tumor doing in the meantime? It's creating millions more cells. You've lost the race. And Steve Rosenberg understood this many years ago and did a really interesting study, experiment, and treatment, where he isolated from tumors the killer cells that were trying to kill the tumor. They're called tumor infiltrating lymphocytes, TILs. He isolated those cells, he grew them up in bathtubs full of IL-2, and then he infused them back into the patient. And if you'd seen Steve give a talk 15 years ago, he usually had a same series of slides, was a guy who had a melanoma metastasis in the lung that was the size of a softball. And he isolated TILs, he grew them up, he infused them back in, and that tumor went down to the size of a baseball. And then he did it again, gave him a second treatment, and the tumor went down to the size of a golf ball. So it was working. And he stopped, and the tumor grew out and killed the patient. Now, why did he stop? Because he's stuck in the self-non-self model. Because he's stuck with the idea that once you turn the immune system on to some antigen, the mere presence of that antigen will maintain the response. And it's not true. You need to continually reboost the immune system. Once a T cell has been activated, its activation, it peaks at about day 8, immune response, and it's gone by about day 14. So he infuses all these activated killers into this guy with a tumor. They go and kill all the tumor cells they can kill, and then they rest, and they need to be reactivated. So he does it again. They go and kill some more tumor cells, and then they rest, and they need to be reactivated. And he doesn't reactivate them. And this, I think, is the problem with almost all the immunotherapies out there. Vaccines against tumors, people are in what I call vaccine mode. They're blinkered. They think of a vaccine, you prime, and then you boost, and then you stop. And that works for infections, because when you prime, you activate and expand the number of T cells against, say, flu. And then you boost, and you expand them some more. And then you stop. And a lot of them die, and the rest of them go into what is called a resting memory state. And to be pulled out of that resting memory state, you need alarm signals. And when the flu comes in, you will get alarm signals. And so the prime boost works because the damage done by the pathogen restarts that primed response. Good so far? Right. It doesn't work for a tumor because the killing done by a killer induces programmed cell death. The immune system does not throw fuel on the fire. So you activate the killer cells. Let's say you vaccinate. You activate the killer cells against the tumor. Takes 3 days. 4 days, they leave the lymph node. They go and kill the tumor cells. That killing does not give you alarm signals. So it doesn't boost the response. And that's not theory, that's fact. That was done by Bill Heath in Australia. The killing by a killer induces programmed cell death. The dying cells are picked up by scavengers. They don't release alarm signals. They don't maintain the response. And so these people who are doing tumor vaccination using the same protocol that you would use for vaccination against a pathogen are stuck in, I was going to use a bad word here, the old model. You need to keep boosting. If you have a vaccine that makes a tumor get smaller, you need to keep boosting. And they're not doing it.
Or you could induce damage in that area in some way.
You could. Oh my goodness, you could. Stick a needle in it. Irradiate it. Right? Irradiation will induce an immune response because usually you get so much damage that you, even though irradiation causes programmed cell death, that you overload the local scavenging capacity. Okay? You stick bacteria in a person's bladder that has bladder tumor. What are you doing? You're turning on your local APCs. Keep doing it. Okay, now let's get to PD-1 and PD-L1, right? Quote unquote checkpoint inhibitors. In order to inhibit a response, or in order to inhibit the tissue from inhibiting a response, which is what they're trying to do, right? They're assuming that your immune system is already turned on to that tumor. And now they're going to block the tumor from inhibiting that immune response, right? Who says your immune system is already on to that tumor? There's nothing to say your immune system is already reacting to that tumor. Is that tumor sending alarm signals? Not necessarily. You need to induce an immune response to that tumor before the PD-1 and PD-L1 inhibitors are going to work. And you need to keep inducing that response. And a long time ago, Tom Waldman was one of the people who invented that, asked me, how can I make this better? And I said, induce an immune response first. I'm out in the sun a lot. I train, sheepdogs and so on. And I get these little basophiliomas on my nose. And I'm also nickel sensitive. So I go and buy a cheap pair of earrings. I break off the part that goes through the ear. I stick the flat part to my nose. It induces a delayed type hypersensitivity response, the same as poison ivy. And the tumor goes away. I'd say, if you have a treatment that makes a tumor get smaller, don't stop. And if you're using something like a checkpoint inhibitor, make sure you've got an immune response first.
So you would expect people who received PD-L1 after surgery on the tumor to have more of a response than those. I'm just thinking out loud.
Yes, but yes, absolutely. But it would be temporary. And you have to make sure that you didn't take the whole tumor. I mean, the thing is, you need the antigen. You need the alarm signals, right? I mean, there used to be these, examples in the literature of, in the older days, especially, of somebody who goes and takes a tumor out, but can only get part of it, and the rest disappears. Well, when you only get part of it, you've done damage to the other part, right? Because you cut it. Right? You've got alarm signals, you've got the antigen, you got to turn on an immune response. And if what you have left is small enough that up peak at day 8 and down by day 14 is enough to kill all those tumor cells, great. If you leave 10 cells, that response is going to come back. You ever heard of Coley's toxin? Bill Coley in the 1890s was curing, let's use the word, 30% of his sarcoma patients by injecting nasty bacterial toxins into their tumors repeatedly. I don't think, for many tumors, we're not doing much better than that today. So I think we have to stop thinking that a tumor is immunogenic just because it's a tumor, because it's just a healthy cell that's lost growth control. And we have to stop thinking that by doing a treatment like a vaccination against a tumor once or twice is going to be enough. So that's how the model deals with it. There's more. I mean, there's lots more complex stuff, but that's the basics.
Interesting. I think we only have time for maybe one more area. There's so many possible territories that this model covers, but I want to talk just at least a little bit about autoimmunity. So how does the danger model account for autoimmunity? And then also I'm just sort of curious your thoughts on like why the joints, and why are women more likely to have certain autoimmune diseases compared with men?
Oh goodness. So the basic question that has been asked about autoimmunity for a long, long time is, how do we break tolerance? I think it's the wrong question. I think we're never tolerant. I think the tissues are constantly inducing tolerance to themselves. But as the body changes, the tissue change. And so you will have, at any one moment, some T cells and some B cells that are autoreactive. Bone marrow is making millions of new B cells a day. The thymus is making millions of new T cells a day. Fewer as you get older, but still some. You're never tolerant. So how can you turn on immune responses to yourself? One, you can have a pathogen that has a molecule on it that looks like a molecule that you have. Rheumatic fever. Kids who make a response to strep. Most of them clear strep without a problem. A few have a molecule on their heart, usually the valves, that looks a little like a molecule on strep. And so while they're clearing the strep, they're also destroying their heart. You put them on antibiotics, the strep stops, the autoimmune response stops. That's important because it says that the mere presence of the antigen, in this case, your heart, is not enough to maintain the response. Okay? You need the pathogen. You need the damage. And that's important for cancers, right? You need to keep reducing that response. That's one way of getting an autoimmune disease. Another one is that you have a pathogen in the place and you don't know it. For a long time, for not that long, but for some time, Lyme disease was diagnosed as juvenile rheumatoid arthritis because we didn't know we had a pathogen. And then the pathogen was discovered, and we now know it's not autoimmune. It's really a pathogen. How many more autoimmune diseases are like that? How many arthritides are actually due to a pathogen we don't know about yet? That's two ways of getting autoimmunity. A third one is bad death. And this is where we get to the woman thing. Programmed cell death, normal programmed cell death, where a cell dies, and there's a lot of it in the body, right? Cells are constantly dying in the body. They get scavenged by their neighbors or by special scavengers whose job is to pick up dying cells. And they don't send alarm signals. And there's a lot of genes involved in that. There's a lot of cellular processes involved in that. And if you have a mutation in one of those genes, or if you've got some environmental insult that does something to one of the cellular processes, you're going to end up with bad death. And bad death gives you an immune response. So, my favorite disease here is lupus. And lupus is one of those diseases that is mainly female. But again, God is in the details. Pre-puberty onset lupus doesn't have nearly the sex bias that post-puberty onset lupus does. Now, pre-puberty onset lupus is more rare, but it happens. So why is post-puberty autoimmunity more common in women? And I think it's because there is death that goes on in female bodies that doesn't go on in male bodies. We pop an ovary every month. We kill off, clean up, and regenerate an entire uterine lining every month. That's a lot of death. It's a lot of cleaning up to do. It's a lot of regeneration. It's a lot of genes involved in these processes. So I think that a lot of lupus is either due to a genetic mutation in a death process or an environmental insult that changes one of the processes involved in normal cell death and scavenging. And it fits a little. I started thinking about this, 10 or 12 years ago when I was at UPenn giving this similar talk, and a woman came up to me. She was probably in her 80s, and she said she was diagnosed with lupus at 13. And her GP already back then knew something. He said, if you make it to menopause, it's very likely it'll go away. And she made it to menopause and it went away. And in some lupus patients, it goes away when they're pregnant, when they're not popping an ovary and not changing their uterine lining and so on. Right? And it comes back with a vengeance when they give birth. And in other people who don't have lupus, they can get temporary lupus while they're pregnant, which makes no sense. Unless you think that during fetal development, there's a lot of death, right? In order to make a hand, you kill off the cells in this primitive paw to make the fingers. That's programmed cell death, and they're scavenged by their neighbors. And there's a lot of it going on in fetal development. And imagine that fetus has a mutation, that there's something wrong with its death or scavenging process. It's now sending alarm signals and releasing DNA, RNA, tubulin, SNRPs, all those cells that are dying and not getting scavenged. The antigens you make antibodies to in lupus. You make anti-intracellular molecule antibodies in lupus. It's a hint that there's something going on with cell death and scavenging. So those fetuses usually don't make it. They abort. And after they abort, the mother doesn't have lupus anymore. It's the fetus and its death processes that are driving the lupus in the mother in that case. And we don't have time to do all of autoimmunity. Can I give you a very quick one on allergy?
Absolutely.
So an old question in allergy has been, I can't answer everything in allergy, but I can answer one. An old question has been, what makes an allergen an allergen? Why is it in the house dust mite, with all the proteins that a house dust mite makes, 2 of them are allergenic, DERP1 and DERP2? Why is it in all the lovely things that a cat makes, 1 of those proteins is an allergen, FeLD1? What makes an allergen? So here's my hand-waving response. An allergen is an immunogen. Remember I said there's a difference between an antigen and an immunogen? An immunogen is something that by itself induces an immune response. So I think there are 4 categories of allergens. 1, a molecule that itself does damage. And it turns out that DERP1 is a protease that attacks the surface of lung epithelium and also the surface of B cells. So it does damage. Right? Category 2, you're packaged with something that does damage, like bee venom. Category 3, you mimic an endogenous or exogenous alarm signal. By the way, pollen, which people think is just innocuous dust, right? The pollen grains are covered in proteases that dig through the pistil of the plant, down the pistil of the plant to get to the egg at the bottom, covered in proteases. Dangerous things. Right? Category 4, you are given at a time and a place where damage has already occurred or is occurring. And my favorite here is peanut. Peanut allergy didn't exist until the 90s or so. And if you think about it, I grew up in Holland. We ate peanut butter for breakfast often. All those kids in Indonesia are eating peanuts all the time. Okay? Why is peanut allergy now a problem? So I didn't know the answer to that, but I was in Canada at Guelph, and I can't remember the man's name, but he studies allergy in Canada. And it turns out that he showed that peanut oil in baby creams can give you peanut allergy. So here's the scenario. You have a baby that has a rash. Some babies have a lot of rashes. Parents don't like having rashes on their baby's skin, so they put creams on them. And peanut oil is a wonderful oil. So a lot of baby creams had peanut oil in them. Now you've got a rash, damage, activated APCs, and you're slathering them with peanut oil. Surprise, surprise that you get an immune response. So the anecdote that kind of fits with this is there's a couple at the NIH, an immunological couple. They studied tuberculosis. They had a baby with a lot of rashes, and they knew this. So they went to like health food stores and got baby creams that use sesame oil instead of peanut. And when that baby was 1.5 years old, he anaphylaxed in his daycare when they fed him hummus. He survived. Hummus has sesame in it. So if I had a baby with rashes, I would probably give it mineral oil, right? Which is at least something that you don't encounter all the time in your life, or just live with the rashes. So 4 categories. You are dangerous yourself. You're packaged with something that's dangerous. You mimic an exogenous or endogenous alarm signal, or you're given at a time when you have damage from something else. And there's an experiment I've been asking people who study allergy to do for at least a decade. And that is, my prediction is that if you took children in a family that were not atopic, but there were also children in that family that were atopic, right, allergic, that if you tested the non-allergic kids' immune responses to the allergens the allergic kids were allergic to, the non-allergic kids would be making immune responses. They're just making IgG or IgA instead of IgE. Because that allergen is in their environment. All right, it has to be kids being raised in the same family, right? And they're immunogens, but those kids are also going to be responding. They're just not making IgE. So what I don't know is, why do some people make IgE and some people make IgG or IgA? That's the part of allergy I don't have an answer to yet.
Fascinating idea for an experiment. What are some other experiments that you have in mind that you have a specific prediction, and if it came out one way or another way, it would support or not support your model?
That's a really good question. So, all right, here's an autoimmune prediction. If you're a rheumatologist and you get girls coming in their teens with lupus, put them on the pill and don't let them cycle, and see what happens. It's called, diagnosis by treatment. I would predict that for a good percentage of them, they would stop having lupus symptoms. I can't get anybody to do that. I can't get them to do that. And it makes no sense to me. They say, oh, that's not normal. You're supposed to cycle. And I say, no. We didn't evolve to cycle every month. Before the pill, women of childbearing age were either pregnant or lactating most of the time. They have done the study of putting them on the pill, but they let them cycle. So that's, yeah. I would like Steve Rosenberg to keep giving TILs. Don't just do it twice. If your TILs work and they make the tumor get smaller, do it again. I asked him why he didn't do that. He actually gave a talk. Where at the NIH, where he happened to let slip that he had given 2 patients their TILs 3 times. And both of them went into remission. And so I said to him, why don't you do that more often? And his answer was interesting. He said, I only have so much money. I only have so many clinical fellows. I only have so many people that are willing to give me their bodies. I want something that's going to work in 2 shots. And I said, ain't going to happen. It ain't going to happen. Do it again. So those are 2.
Out of curiosity, how did your colleagues react to your original proposer of the danger model, and how has that sort of changed over time? So
originally it sank like a stone. I mean, it was an annual reviews paper. It was very long. It was mostly about tolerance. And then we redid the experiment Medawar had done that put the self-non-self model on the map, for which he got a Nobel Prize, and showed that he'd misinterpreted his data. 3 labs back to back showed that you could immunize baby mice without a problem, that they were not simply going through a period of learning self. And actually that rebirthed a whole bunch of research on neonatal vaccines, which is kind of nice. I actually had an impact on medicine once. And then the shit hit the fan. So I remember in Australia, there was actually a newspaper article with the title, Nobel Prize to be retracted, ? And I was just going down there to give a talk, and oh my God, were the talons out. So the first thing, it sank like a stone. The second thing was, can't possibly be right. It's total bullshit. Nobody's going to believe this, da, 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 da. And then we did the experiment to show that necrotically dead cells can act as adjuvants, that you can use them to induce an immune response. And then people started getting interested. But what's happened today? This is where it is today. There's this whole pie that the danger model explains. And people have accepted one small slice of it. So people tell me, I teach danger. I said, great, what do you teach? And they tell me, well, when we're during the chapter on activation of APCs, antigen-presenting cells, we talk about PAMPs, pathogen-associated molecular patterns that activate them, and DAMPs, damage-associated molecular patterns, right? And so there are internal things that can activate your APCs. I say, great. What do you teach about why mothers don't reject their fetuses? Oh, it's fetuses immunosuppress their mothers. So here's what has happened, is they've accepted, because there's so much evidence, that there are internal alarm signals that activate APCs. But they haven't applied that to all the rest of immunology. For all the rest of immunology, they're still stuck in that old model. So I don't know what it's going to take. That this part, I mean, if you accept one basic assumption, and then you don't then ask, what does that assumption imply for the rest of it? Where's the logic? Where's the thought process? Where is the rigor of science that you're not willing to change your assumptions for the rest of it? So that's where it is today, pretty much.
What do you think of that recent discovery that some very, very large percentage of people with MS have EBV compared to the general population?
Well, 90% of the general population has EBV. I have long thought that MS is more than one disease. And we know that from the treatment, right? Some people respond to interferon beta, some people don't. So in mice, there's a virus that if you give it to the mice, it goes to their brain and it very much looks like MS. Yeah, I first heard about EBV, must be 15 years ago, when a guy put EBV together with the finding that people in the Northern Hemisphere get more MS than people in the South. Well, it turns out it's not people in the Northern Hemisphere. It's people who grew up in the Northern Hemisphere. And not only that, it's people who were in the Northern Hemisphere between the ages of about 12 to 22. If you then move south, you're going to get MS. And if you move north after that, you're not going to get MS. Well, right? So the incidence of MS is high in people who were in the Northern Hemisphere from 12 to 22. So vitamin D. We know that vitamin D has great effects on the immune system. It's misnamed. It's not a vitamin. It's hormone. Your skin makes it, right? It goes to the nucleus of cells and changes the expression of thousands of genes. And one of the things it does with the immune system is it tends to give you, if your vitamin D sufficient, it'll tend that you will now make an immune response which is not as inflammatory as if you were vitamin D deficient. So it's not that it's damping the response, it's shifting it from a highly inflammatory poison ivy kind of response to a different kind of response. Still very effective. Okay, so EBV is kissing disease. 70% of freshman college students don't have it, and 90% of seniors that are leaving college have it. Most of us never know we get it. Few people get mono, not many. So here was his idea, that if you are vitamin D sufficient at the time that you get EBV, you will make a not very inflammatory response to EBV. If you're vitamin D deficient at the time that you get it, you will make an inflammatory response against it. That was his idea. 90% of the world has EBV. 95, I think. So now we add that to the fact that you can find EBV in the brain. And there you have the EBV, the Northern Hemisphere, and the vitamin D all put together. Isn't that cute? Not my idea. It was very cute. I don't think EBV is going to be the only one. I have a friend who's a veterinarian who got MS when he was 23, and he says a lot of vets get MS. And he, because he has access, has kept himself on antivirals ever since. He's now 80-something, and he walks with a cane. That's as far as his MS has gotten. Antivirals all of his life. So I wouldn't be surprised if it's not just EBV. We have retroviruses that can activate. We have the potential to have a gut bacterium, which has a molecule that mimics a molecule in the brain. We now know this for a form of autoimmune uveitis, that there are bacteria in the gut that have on their surface or inside, I don't know, a molecule which looks like a molecule on the eye. And as long as you're fighting off those bacteria, you're also destroying your eye. It's a little like rheumatic fever. So there could be that form of MS as well.
And on that great note, thank you so much for being on the External Medicine podcast. For people that are interested in learning more about you, where would you have them go on the internet? We'll definitely include the lecture series that you did at the NIH, the 8 lectures, in our show notes. Is there anywhere else you would have them go on the internet?
The BBC did a 1-hour program. I can't remember if it was called Horizon or Nova. You know, the Horizon series. It's called Turned On by Danger. It's old. It was done in 96. Yeah, isn't that a great title? You'd think it was about a race car driver or something. It's old, but it does a pretty good job of explaining the danger model for non-biologists even. No, I mean, I would think if they're interested in me, then the Wikipedia has a page on me that I check it about once every 6 months to make sure that nobody's lying. They made a mistake. They said I published with my cat, and I had to fix that and say, no, it was a dog. Every so often somebody, about a year ago, a woman named Katlin Carrico, who you should also interview, told me that she'd seen my 8 lectures and she sent me her notes that she still had. Do you know who Katlin Carrico is?
Yeah, mRNA.
The mother of the mRNA vaccine. Yeah, that's an alarm signal. It's about time that we started making vaccines using alarm signals instead of the adjuvants that make so much damage that they elicit them. Yeah, good for her. Hope she gets a Nobel Prize soon. Anyway, I really enjoyed this. That was really fun. And I really I commend you on doing what you're doing.
Well, we're only as good as our guests, and we lucked out with having a really great guest tonight. So thank you so much.